Hey guys, welcome back to my channel. Today I am going to continue with the medicinal chemistry 4th semester 5 marks question and answers from unit 1. So the 10th question is explain the role of chelation and bioisostereomerism, and the answer is the role of chelation. It is a complex of drug molecules that can't cross the natural membrane barriers and they render the drug into the biological ineffectivity. The rate of absorption is proportional to the concentration of the free drug molecules, that is, the diffusion of the drug. Due to the reversibility of the complexation equilibrium between free drug and the drug complex, which gives drug plus complexing agent, which gives or yields the drug complex. So the complexation reduces the rate of absorption of the drug but not the effect of the availability of the drug. Chelation is a type of bonding that involves in the formulation of two or more separate coordinate bonds between the polydentate ligand and a single central metal atom. So the importance of chelation in pharmaceutical medicines are 8-hydroxyquinoline and its analogues act as an antibacterial and antifungal agents with complexing of iron and copper. So the side effects caused drugs which chelates and metals. Phenobarbital forms a non-absorbable complex with polyethylene glycol 4000 and the calcium with EDTA form a complex which is increased the permeability of the membrane. And the role of bioisostereomerism. The bioisostereomerism is defined as a compound or a groups that possess near or equal to the molecular shape, size and volumes approximately the same distribution of electron and which exhibits the similar physical properties. So the bioisostereomerism is classified as two types. Number one, classical. Number two, non-classical. So the classical bioisostereomerism, they have a similarities of shape, size and electronic configuration of atom, group or a molecule which they replace. So the classical bioisostereomerism may be univalent, bivalent, trivalent, tetravalent or a ring equivalent. So the non-classical bioisostereomerism. The non-classical bioisostereomerism are the functional group with dissimilar valence of an electronic configuration. They do not obey the steric and electronic definition or the electronic configuration of a clinical isostereomerism. The specific characteristics of non-classical bioisostereomerism are electronic properties, physicochemical properties of the molecule, spectral arrangement, functional moiety for biological activity, example halogens and ether. So question number 11, explain optical and geometrical isomerism in relation to the biological action. The optical isomerisms, when the two molecules only differ with three-dimensional position of the substituents around one or more atoms, they are called as optical isomers and the phenomenon is called as optical isomerism. The stereochemistry, enantiomers, symmetry and the chirality are the important concept in therapeutic and toxic effect of the drug. A chiral compound that containing one asymmetric center has a two enantiomers, although each enantiomer has identical and chemical physical properties. They may have different physical activity like interaction with receptor, metabolism and protein binding. And optical isomers in biological action is due to one isomer being able to achieve a 3 points that are attached with the receptor molecule, while the enantiomers would only be achieved 2 points attachment with the same molecule. The category of drug where the two isomers have qualitatively similar pharmacological activity but have different quantitative potencies. So the geometrical isomerism, it is a form of stereochemistry or describing the orientation in the form of functional group within a molecule. So the geometrical isomerism is a type of isomerism that arises in the heteroleptic complexes due to the different possible geometric arrangements of the ligands. So the important examples of these behaviors are found with coordination number, that is 4 and 6. There are two types or simple types of geometric isomerism which are possible for the octahedral complexes. The geometric isomerism is represented by trans or cis isomerism, so the resulting from restricted rotation due to the carbon-carbon double bond or in the rigid ring structure or system. So question number 12, discuss hepatic and extrahepatic metabolism. The answer, hepatic phase 1 biotransformation. Refer question number 1 for biotransformation. I am going to continue with the answer. Number 2, reduction. So the reductive reaction which usually involves in the addition of hydrogen bond to the drug molecule, which occur less frequently than the oxidative reaction, and the reduction enzymes are responsible for the reduction of xenobiotics that requires NADPH as a cofactor substrate for reductive reaction, which includes azo or nitro compounds, exopoids, heterolytic compounds and halogenated hydrocarbons. So the biotransformation by reduction is also capable of generating a polar functional group such as hydroxy and amino groups, which can undergo for the further transformation. Hydrolysis, esteramides, hydrazides and carbamates can be hydrolyzed by various enzymes. The hydrolytic enzymes that metabolic drugs are the one that act on the endogenous substance that are found in many organs like kidney, intestine, plasma, etc. are a number of drugs with ester, etheramide and hydrazide linkage undergo the hydrolysis. The important example are cholinesterase, procaine, procainamide, etc. So the reaction of the hydrolysis. Extrahepatic metabolism. So the extrahepatic metabolism, refer question number 2, phase 2 biotransformation. I am going to continue with the answer. Number 3, conjugation with sulfate or sulfation. So conjugation with sulfate is similar to the glucuronidation, but it is catalyzed by non-microsomal enzymes, whereas the glucuronidation is catalyzed by microsomal enzymes and less occur commonly. So the conjugates of sulfates, referred as a sulfate ester conjugate or ethereal sulfates, that unlike the glucuronide conjugation, so sulfur conjugation in mammals is less important because PAPS, that is 3-phosphodiosine or 5-phosphosulfate donor that transfers the sulfate into the substrate, which is easily depleted. The drugs undergoing sulfate conjugation, which includes chloramphenicol, phenols and adrenal and sex steroids. So the fourth one is conjugation with methyl group or methylation. This involves transfer of methyl group from the cofactor, that is COA, cofactor S-adenosyl methionine SAM to the acceptor substrate by various methyl transferase enzyme. So the methylation reaction is lesser important for the drug, but it is more important for the biosynthesis. Example, adrenaline and inactivation, histamine of endogenous amine. Occasionally the metabolites formed are not polar or a water soluble and may possess equal or greater activity than the parent compound. So the example is adrenaline is synthesized by non-adrenaline. Fifth one, conjugation with glutathione and mercaptoric acid formation. So the conjugation with glutathione and mercaptoric acid formation is a minor but important metabolic pathway in the animals. So glutathione is a tripeptide that having glutamic acid, cysteine and glycine. It has a strong nucleophilic character due to the presence of SH group in its structure. Thus, it conjugates with the electrophilic substrate a number of potency that toxic compounds and protects the tissue from their adverse effect. The interaction between the substrate and the GSH is catalyzed by the enzyme glutathione S transferase, which is located in the soluble fraction of the liver, which homogenates, and the glutathione conjugates either due to its high molecular weight that is excreted as such in the bile or is further metabolized to form a mercaptoric acid that conjugates and that excreted through the urine. Number 6, conjugation with thiosulfate. It is important reaction in the detoxification of cyanide, conjugation of a cyanide ions, which involves the transfer of the sulfur atom from the thiosulfate to the cyanide ion in the presence of enzyme endogenase to form an inactive thiocyanide. So the thiocyanide was formed is much less toxic than the cyanide and is excreted through the urine. Question number 13. Explain the role of solubility and protein binding. So the answer is solubility, refer question number 7, and for protein binding, refer question number 8. Explain the role of partition coefficient and hydrogen bonding. For hydrogen bonding, refer question number 8, and for partition coefficient, refer question number 9. So question number 15, explain the role of ionization and bioisostereomerism. For answer, refer question number 7, and for bioisostereomerism, refer question number 10. 16th, question number 16th, explain the role of ionization and chelation. So ionization, refer question number 7, and chelation, refer question number 10. Explain the reductive and hydrolytic drug metabolism with their importance. So for this answer, refer question number 12. Explain the role of solubility and hydrogen bonding. Refer solubility as a question number 7 and hydrogen bonding as question number 8. So here we finished the 5 marks question and answers for first unit. Tomorrow I am going to continue with the 2 marks of first unit. So please do like, share and subscribe to my channel. Thank you very much.